Hello. Hi, Ronak. How are you? You are good. That's nice. So, how was your day? Was it good? Okay, that's great. Good evening, sir. Good evening, ma'am. How are you? I'm also good. Thank you. So, my name is Rishikant Vaibhav. I am biology tutor over here. I have done my graduation in life science from Delhi University. I have done my master's in botany from Patna University. In the meantime, I have also qualified for CSIR NET and GATE examination. Yeah, these are national level examination and conducted all over the India. Yes, they are highly competitive and difficult to crack, but with some dedication and hard work, anyone can crack it. Yeah. So, Ronak, do you know why we are here today? Yes, to study about plant growth regulators. So, before I start this, I just want to ask: Have you read any particular book or watched any particular video about it? Okay, it means you like to read books, Ronak. Okay, that's nice. I also like to read books. Yeah. So in this particular session, we will study about plant growth regulators. We will study what are they. We will study about their function, and we will study about why are they important. Important. Okay. Can you tell me one any specific reason why are they important? No problem, Ronak. You will get to know this in this particular session itself. Okay. So let's move on to the topic. So
Yeah,
plant growth regulators, we can call them as plant hormones or phytohormones, but I call them secret messengers of plants. Okay. So Ronak, have you ever wondered why do plants bend or why fruits ripe or why do leaves fall? Okay. Yes. Seasons. Seasons are one of the factor, but they are external factor. What happens internally in plant is hormones, plant hormones regulate this kind of activity internally. Okay, and that's uh, why plants hormone are very important topic to study. So shall we move to the topic now? Okay, let's go then. First of all, we will know what are plant growth regulators. As I have told you that they are also called as plant hormones or phytohormones. So phytohormones are biochemical messenger produced by source cell regulate cellular physiology in target cell through signal transduction pathway. And these hormones include auxin, gibberellin, cytokinin, ethylene, and abscisic acid. Beside these five major hormones, there are some other hormones like brassinosteroid, jasmonic acid, salicylic acid, stigolactin. These also regulate cellular physiology of plants, physiological process of plants, but we will study them in another session. Today we will discuss about only these five. Okay. So now let's move on to the auxin. Shall we move? Okay. So auxin, we call it as growth hormone. It was discovered by Charles Darwin and his son Francis Darwin. They were performing an experiment and they observed that the tip of grass coleoptile senses light and sends a chemical messenger or signal that causes bending in the plant. And later on, this chemical messenger was called as auxin. Okay, so auxin is derived from tryptophan amino acid. Tryptophan amino acid. It can occur in natural as well as synthetic form. Natural form of auxin includes IAA and IBA. IAA is indole acetic acid. IBA is indole butyric acid. Synthetic forms include NAA and 2,4-D. NAA is naphthalene acetic acid, whereas 2,4-D is 2,4-dichlorophenolic acetic acid. Now, this 2,4-D is used in agriculture Ronak, you tell me, what is the purpose we use 2,4-D? For what purpose you use 2,4-D in agriculture? Yes, it is used as weedicide. Nice. Nice, Ronak. That's nice. Now we have gone through the background of auxin. Now let's see the key function of auxins. So key functions of auxins include cell elongation and growth. Uh, Auxin promotes apical dominance. It induces parthenocarpy. It delays senescence. It promotes phototropism, thigmotropism, geotropism. It also promotes flowering in pineapple. Sometimes it promotes vascular differentiation. Sometimes it also take part in sex determination in some families like Poaceae and Cucurbitaceae. In Poaceae, it promotes femaleness, and in Cucurbitaceae, it promotes maleness. Okay. Do you know about apical dominance or parthenocarpy? Okay, no problem. I will explain it to you. Suppose, first of all, let's see apical dominance. Suppose this is a plant. This is a plant, and this is its apical bud. Okay. What happens in apical dominance is this apical bud will continue to grow, or we can say plant will continue to grow only from apical bud. Plant will grow only longitudinally. Now, how auxin achieve this apical dominance? So what auxin does is, there was supposed to be lateral buds also in plants, but what auxin will do, it will inhibit them. It will not let them grow. That's how the plant will grow only longitudinally, and that's how and the apical dominance will be achieved. Do you know about parthenocarpy? Yes, yes, fertilization. A fruit without fertilization. Yeah, fruit without fertilization. Now let's move on to the next hormone, which is gibberellin, the elongation expert. It was discovered by E. Kurosawa while studying foolish seedling or Bakane disease in rice. Bakane disease was caused by Gibberella fusicori. Fusicori. Gibberellins are derived from terpenes. Terpenes are made up of isoprene. Isos. Well, one second. Isoprene. One isoprene is made up of 5 carbon, and two isoprene combined together to make a terpene, two molecule of isoprene. So terpene is technically 10 carbon compound. Okay. Now over the time, hundreds of different gibberellins have been identified, but only GA1, GA3, GA4, and GA7 are the active forms of gibberellin. Now let's see the functions of gibberellin. Okay, gibberellin perform elongation, stem elongation. How does it elongate stems? So it increases the internodal lengths. Okay, and that's how it increases the plant height also. Next function of gibberellin includes germination of seeds. Okay, so it activate hydrolytic enzymes that breaks the seed dormancy and makes the seeds germinates when the time favorable times come. Okay, now next is bolting. Next function of gibberellin is bolting. Bolting is something like sudden increase in length at the time of flowering. So, gibberellin induces bolting in rosette plants, and that's also promote flowering response. Rosette plants includes like cabbage or beetroot also. Other functions of gibberellin includes like it also induces parthenocarpy, and I have also discussed this seed break seed dormancy here also already. It induces pollen grain development. Pollen grain development means it favors maleness in plants. Okay, it delays senescence. It also promotes flowering. Now let's move on to the next hormone, which is cytokinin, the division director. Okay. Before I start with cytokinin, I just forgot to mention one thing about gibberellin. Like gibberellin is derived from terpenes, right? But it is made up of two molecule of terpenes, which is diterpene. Diterpenes. Okay. And diterpenes means it is 20 carbon compound. So gibberellin is made up of 20 carbon compound. Okay. Now let's move on to the cytokinin. Cytokinin discovered by Skoog and Miller. They are adenine derivative. Zeatin is one of the natural form of cytokinin, and one form is kinetin, which is synthetic. Cytokinin naturally occurs in region of rapid cell division, which is RAM. One such region is RAM. Do not get confused this RAM with your laptop RAM. Laptop RAM is different. That is random access memory, and this is root apical meristem. Okay, root apical meristem. Now let's see the function of cytokinin. Function of cytokinin includes it inhibit apical cell dominance. How does it inhibit apical cell dominance? It stimulates the growth of axillary buds and branching. That's how it inhibit apical dominance. This is completely opposite to what auxin do. Auxin promote apical dominance by inhibiting lateral bud growth. Okay. The second functions of cytokinin, it takes part in chloroplast development. That's how it enhances the function of chloroplast, and as well as photosynthetic efficiency of plants. It delays senescence, it prevents premature leaf aging, and it promotes shoot development in plant tissue culture. Now, how does it promote shoot development in plant tissue culture? Uh, you must not know in very detail about plant tissue culture, but let me tell you. In plant tissue culture, what we do is take a part of plant which we call callus, any part of plant which we call it as a callus. Now, this callus will be provided with favorable environment and suitable growth media, and this callus will, it might, depending upon its property, it might develop into a whole plant or a part of plant. Okay. Now, what happens in growth media? This cytokinin is present in the growth media itself. Depending upon the concentration of cytokinin and auxin, the it either it will promote shoot development, or either it will promote root development. So if cytokinin level is more than auxin, it will promote shoot development, and if cytokinin level is less than auxin, it will promote root development. So this is about cytokinin. Now let's move on to the next hormone, which is ethylene, the gaseous regulator. Ethylene is only gaseous plant hormone. Okay, it is derived from methionine, and it acts as both as growth promoter as well as inhibitor, depending upon the concentration, and tissue type. Ethylene play important role, like it promotes fruit ripening, and it what does happen in fruit ripening? It triggers some enzymatic changes that leads to change in color. Like in figure you can see the unripened plant is green and ripened banana is yellow. So this all is because of ethylene, and it also changes the texture and flavor of that plant. Like the green banana would be hard, but the ripened banana would be soft and sweet, which is not present in the unripened banana. So second function, it promotes senescence, like it accelerates the aging process in whole plants, either leaves, fruits, and flowers. In whole plants, it accelerates the aging process. Third is it shows triple response. Before I come to the triple response in detail, let's see the fourth function. It promotes epinasty leaf development. What happens in epinasty leaf development? The petiole of the plant, which connects the leaf to branch, it gets elongated, and the leaf sometimes bends like that. So it captures more and more sunlight, and that's how its photosynthetic efficiency increases. What now comes to the triple response? What happens in triple response is that ethylene inhibit the shoot elongation. At the same time, it promote the stem thickness or shoot thickness. Okay. uh, and third, what it does is that it seizes tightens the apical bud, so that apical bud ceases to grow. That's how the triple response of ethylene is done. Now we are done with ethylene. Let's move on to the last but not the least hormone, the abscisic acid, which is we call it as stress manager. So the abscisic acid is also known as stress hormone, and it is synthesized in almost all cells which contain chloroplast, and it is derived from zeaxanthin. Zeaxanthin is a 40 carbon compound. We call it as tetra terpene. Okay, and now it acts as growth inhibitor, and it also acts as ghrelin antagonist. So functions of abscisic acid includes stomatal control. I will come to that after all the functions. It maintains dormancy. It primary function. It prevents premature seed germination and maintains bud dormancy. What happens? What is the benefit of this? That seeds will grow only when the conditions are favorable to germinate. And that's how it helps the plant. Now, the third function is it induces abscission. It induces abscission in leaves, fruits, and flowers. It delay flowering. It inhibit SOC, which is a master regulator. SOC is a gene which is a master regulator of flowering. Master regulator of flowering. Now it induces root development and inhibit shoot development in drought condition. What happens in drought condition is like there will be lesser amount of water, or water table will be much below than the expected level. So in drought condition, abscisic acid will tend help in root development, so that roots can go deeper to find the water. At the same time, it also closes stomata. That's why I left it for the last function. It closes stomata so that no transpiration can occur. No transpiration will occur. Okay. And that's why we also call it as anti-transparent hormone. No transpiration will occur. So what will happen is no loss of water. So it is the last thing any plant would like to do in water stress condition, to lose their water. Okay. So now we are done with the hormones, all the five hormones. Auxin, gibberellin, cytokinin, ethylene, abscisic acid. Now let's do a quick question. Okay, so Ronak, tell me, first question, which hormone maintains apical dominance? Yes, great. Auxin. Auxin maintains apical dominance, and how does it do? It prevents lateral bud growth so that apical dominance takes place. Now, second question is, bolting in rosette plant is induced by? Yes, gibberellin. Yes, right. Last, third question, the stress hormone. The stress hormone of plant is, yes, abscisic acid, right? Manage, as I have told, it manages water stress much way better. So now we are done with question. I have added a summary of all the hormones, not all the hormones which we studied today, like auxin, primary function, cell elongation, apical dominance, tropic response, gibberellin, stem elongation, seed germination, bolting, cytokinin, cell division, delay aging, shoot development, ethylene, fruit ripening, senescence, stress response, abscisic acid, dormancy maintenance, and stomatal closure. Now, one thing I would like to tell that all these hormones will work together in a coordinated way so to give their effect. I might have written that here. Yeah, each hormone works in concert with other, creating complex regulatory network that control plant development and response to environment stimuli. Okay. So that's it for today, Ronak. I hope you like the session. If you like the session, you will have to give us the rating. Yeah, rating is very important to us. So how was the session according to you? Was it good? Okay, that's great. You like the session, Ronak. Now I would request you to call your parents, either mother or father. I just want to talk about session. Yeah. Hello, sir. We just took a session with Ronak and it was a great session. He is very intelligent and he's very curious. Yeah, he just wants to learn all the things, and he gave answers to my questions, which shows he is very focused in the class while studying. But we'll have to work with him more diligently on his basics, because you know, sir, clear understanding of basics will make advanced studies easier for him. Yeah, for that I will be regularly monitoring his revision, and I will be giving him practice questions also. Yes, sir. Yes, sir. So I just want to know from you also, sir, what is your expectation from me and organization? Yes, sir. We will work with him definitely. I will give him worksheets, homeworks, assignments, all. Yeah, yeah. This will help him in getting clarity. Yes, definitely, definitely. And he's, by the way, he's doing great and he will do wonders with our help. Yeah. Right. Yes, sir. Rest assured, sir. We will give him worksheet, we will give him assignments, we will give him homeworks, we will take test of him, we'll do all sorts of things which is required. Yeah. Thank you. Thank you, sir. Thank you. You like the session. Thank you so much, sir. Yeah, sir. Yeah. I will definitely, yeah. The discount which you are talking about, I will convey it to the respective department. Yeah, there is discount. I will convey it to them. They will call you back just after this session. Yes, sir. Yes, sir. Don't worry about that. They will call you after that. Okay. Yes, sir. Thank you, sir. So the session was quite good. It was a interactive session. So Ronak, I am leaving this with a great quote. Just study, just stay curious and stay prepared, and we will crack all the exams. Thank you.